Can you talk about your professional background and how did you get involved in the psychedelic research field?
Okay, my name is GÜL DÖLEN, and I am a professor and Parsons Endowed Chair at the University of California, Berkeley. I'm also in the Department of Psychology and the Helen Wills Neuroscience Institute there. I also maintain an adjunct professorship at Johns Hopkins University in the Department of Neurology and Neuroscience. I started my lab about 10 years ago at Johns Hopkins, and I just recently moved the lab to Berkeley this past year.
Could you explain us the neurobiological mechanisms of how psychedelics work and how do they affect the brain?
The discovery that my lab made is that psychedelics seem to be what we call the master keys for unlocking something called critical periods. So critical periods are these windows of time where the brain is especially sensitive to its environment and able to learn in lasting ways from that environment. So most people will be familiar with the notion of a critical period when they think about how hard it is to learn a second language as an adult compared to how they learned their first language as a child. And that's because there's a critical period for language learning, and as you get older, it closes. And so this has been something that neuroscientists have known about for almost 100 years. And we've had this insight that if we had some kind of mechanism or some understanding of why critical periods close, that we could use that information to find new ways to reopen them. And the idea has been that the reason that we're so bad at curing diseases of the brain is because by the time we get around to intervening, most critical periods are closed. And so if there was a mechanism or a master key for unlocking critical periods, we've had the intuition that we would be able to cure diseases of the brain much more easily. And despite the fact that we've known about them for about 100 years and that 3 Nobel Prizes have been awarded for understanding their mechanism, really I think we have been not making that much progress on finding these master keys. And so discovering that psychedelics might be these master keys that we've been looking for is a sort of breakthrough, because at the same time as the neuroscience community has been hunting for the master keys, the psychedelics community has been discovering that psychedelics drugs are these compounds that are curing such a wide variety of disorders. And it was hard to kind of explain why it is that despite the diversity of the different diseases, that psychedelics seem to be working in so many different ways. And so bringing the two ideas together, if psychedelics really are the master keys for unlocking critical periods, it would explain why they're also effective in so many different disorders of the brain.
So psychedelics are the master key, but what do they do in the brain?
What my lab has discovered is that the way that the psychedelics seem to be working is that when they bind to the receptors that, there are a lot of different receptors, a lot of different psychedelics, and that when that happens, it triggers a set of cellular changes that causes the extracellular matrix, which is this protein-sugar complex that surrounds the synapses. It causes that to dissolve, and that enables the cell to reorganize its synaptic weights and restores plasticity to an adult brain. So that phenomenon of restoring the ability to induce plasticity is something called metaplasticity. And so this degradation of the extracellular matrix plus the metaplasticity, we think, are the mechanisms underlying this ability of psychedelics to reopen critical periods.
Can you explain us the term plasticity in this context?
So synaptic plasticity is a term that's been widely used in neuroscience, and different people mean different things by it. But as a synaptic physiologist, what I am referring to is the change in the response properties to a specific stimulus. So if you give a stimulus and then you measure the response to that stimulus, and then you do some manipulation. For example, if you want to give a peptide or change in the electrical pattern of stimulus that you're giving that can induce plasticity, then the same stimulus will give a different magnitude of response. And so that change is what we call synaptic plasticity, and it's been proposed that that synaptic plasticity is the basis of learning and memory.
Is there any evidence that psychedelics can induce neurogenesis as well, like creating new cells?
So that study was not really reproduced. It's not clear that they do that. In fact, I would just, I think there's a lot of enthusiasm for the idea of growth and plasticity as being the mechanism of psychedelic. But I just want to caution that not all growth is good growth and not all plasticity is good plasticity. It's the same principle as in cancer, right? Not all growth, if it's not developmentally regulated in the appropriate way, sometimes too much growth, sometimes too much neurogenesis, sometimes too much plasticity means cancer, right? And so similarly, too much plasticity has been associated with drugs of abuse. And so we're very careful in how we describe psychedelics. And what we're finding is that they don't just induce plasticity. What they do is they restore the ability for stimuli to induce plasticity, right? And the difference is that it's the difference between taking a pill and tomorrow being able to speak Japanese, or taking a pill and tomorrow being able to learn to speak Japanese. And those are functionally and mechanistically very different phenomena.
In the media, we often see colorful brain scanning images, but can we really relate or connect those images to the very sophisticated experiences people have under the influence of psychedelics?
Yeah, I have to say that in general, our studies in the octopus have really, I think, challenged the view that we're learning as much as we think we are from these types of imaging studies, right? So in the imaging studies that people are seeing, they'll give a psychedelic and they'll say, oh, okay, here's an increase in signal in this brain region and a decrease in this brain region, and that explains what psychedelics are doing. But then you do the same experiment, you give an octopus one of these psychedelic drugs. So we gave MDMA to an octopus. And an octopus, that experiment, the reason that it's so significant is because an octopus does not have any of the same brain regions that humans do. Their brains are so different from ours. They evolved, along totally different evolutionary histories that they don't have a cortex, they don't have an amygdala, they don't have a nucleus accumbens, they don't have a default mode network, and yet they respond to these psychedelic drugs behaviorally in the same way. And so what that's telling us at a really fundamental level is that these anatomical explanations are just accidents of evolutionary history, but the mechanism is something more cellular, more molecular at the level of the things that the two species share.
Can you talk about the rat park experiment and how did it change the way how we think about laboratory experiments?
So the rat park experiment was essentially a challenge to the way that rodent experiments were being conducted to model drug abuse. So mostly in the early days of addiction research in rodents, people were focused on the reward value of the drugs, and they were testing these in animals who were living in social isolation. But rats and mice are actually social animals. And so it turned out that the ability to sort of reproduce this addiction-like behavior depended on whether or not they were living in isolation or living the way that they normally live in the wild in social groups. And so what the rat park experiment did is it restored sort of their normal complexity of social interactions and also learning opportunities and environmental enrichment. And when they did that, they showed that addiction-like behaviors were much harder to induce in a rodent. And that experiment has been used to suggest that, it's not just the abuse liability of the drug itself, but also combining that with the susceptibility to that addiction-like behavior based on the stresses and the environment and the social factors that are important for social animals.
Is it only true for social animals or also for asocial animals such as octopuses?
Yeah, so nobody, as far as I know, has done any, testing in octopuses for addiction-like behaviors. I do think that they would probably feel much less stressed. In fact, they would prefer to live asocially. So, it's basically unknown. As far as I know, nobody's done that experiment.
Most researchers are doing research with mice or rats. Why did you choose animals like octopuses to do research?
Yeah, so we chose octopuses because, they're evolutionarily very distant from us. So it allows us to test theories about how you get complex behaviors from molecules, synapses, and circuits without getting distracted by similarities that might not mean anything. For example, having a cortex or not having a cortex, right? And so that very distance between them really allows you to generalize the rules for how to build complexity.
Aristotle told that octopuses are stupid animals. Do you agree?
No, I do not agree. Aristotle's conclusion that they were stupid was based on the fact that if they're very curious, right? And so if you place a object in front of them, they will grab it, right? And this curiosity made them very easy to catch for, Aristotle lived in a fishing area and people would eat them and hunt them for food. And they were easy to hunt because they could take advantage of this curiosity. I mean, that does not necessarily correspond to intelligence, I think.
So as far as I understand, we can say that psychedelics awaken a curious child in us. Mm-hmm. Is it correct to say that?
I mean, I think that the notion that reopening critical periods is this common mechanism across psychedelics very much is in line with people's phenomenological reports of, feeling like they've returned to a childlike state of curiosity, this sort of appreciation of the world as a child is something that people often describe on psychedelics. So I think phenomenologically it fits.
In the psychedelic fields, many companies are making efforts to find psychedelic drugs without the psychedelic experiences. How do you see that?
Yeah, so our data basically suggests that there is a proportionality between the duration of the trip or the psychedelic effect and the duration of the open state of the critical period. And what these data suggest is that if you are to try and get rid of the trip by shortening that experience down, you will essentially interfere with the ability to open critical periods. And if that's the therapeutic mechanism, then I think that you will essentially eliminate the therapeutic efficacy of these drugs by getting rid of the trip. This is, I think, probably the central debate in psychedelics right now is, do you need the trip? And does the context matter? And I think the way to sort of understand this debate is to realize that for the last 50 or so years, neuropsychiatric treatments have been based on a model of disease that's sort of a biochemical imbalance model of disease. And so if you're depressed, it's because you have a biochemical imbalance in serotonin, and all we have to do to make you undepressed is to restore that balance, right? And I think really what the psychedelics are doing is challenging that entire model of disease, because people are essentially taking psychedelics, having a experience that enables to learn something about how their disease process manifests, change that learned habit that they've built around that injury, and then they are essentially reporting that the disease is cured, right? And it's not 100% cure in 100% of people, but it's a very different model, a learning model of disease rather than a biochemical imbalance model. And I think that the excitement around psychedelics is the idea that we could essentially, instead of having to treat people with a drug for the rest of their lives and only interfere with their symptoms or disrupt their symptoms, that if if we can fine-tune the way that we combine psychedelics with whatever therapy that is relevant for whatever disease that we're trying to cure, then we can essentially start thinking about neuropsychiatric disease the way that we think about, you know, cardiovascular disease, heart attack, stroke, right? Here you do an intervention, you maybe give a few weeks of rest and recuperation or physical therapy if you've had a stroke to relearn what you lost in the stroke, and then you're cured. That's the idea. And I think if that ends up being the case, then this will be a sea change for how we treat disorders of the brain.
What are the new frontiers of research in the psychedelic fields? What are the most exciting questions we would like to answer?
Yeah, so what our understanding of what psychedelics mechanism, this notion that they might be the master keys for unlocking critical periods, really suggests is that even though the clinical focus to date has been on neuropsychiatric diseases like depression and post-traumatic stress disorder, there's a lot of excitement around their ability to potentially cure addiction. This is all very exciting, but what our results suggest is that we might be able to branch out and now address other diseases like stroke, traumatic brain injury, anything that essentially there's involvement of learning that initially encodes the behavior, which is almost everything, because the brain is essentially the reason we have critical periods is that there just aren't enough genes in the genome to encode every single behavior we might need to know, right? So there's not 260 language genes. There are genes that are enabling you to learn new languages. And because you might be born in an environment that's different from what your genetics would encode. And so almost everything is learned that we do that's behaviorally flexible. And so this opens up the therapeutic arena dramatically. And so what my lab is really focused on right now is trying to reopen the critical period for motor learning after stroke, with the idea that if we could demonstrate this principle of, you know, psychedelics being an adjunct that helps learning rather than restoring a biochemical imbalance, then we can start thinking about other diseases as well.
Can you talk about the role of set and setting in the psychedelic experience?
I think the best evidence that we have that the set and setting, or as neuroscientists we call that context, matters, is that when we originally started these experiments, our first paper that was published in 2019, we had made the assumption that the reason that MDMA, which is a psychedelic that has a lot of pro-social properties, was reopening the critical period for social reward learning because of these social properties of the drug. But more recently, what we've shown is that actually all of the psychedelics, whether they have this pro-social property or not, are able to reopen this social critical period. And so that already is telling us that the social part of it is maybe not the unifying mechanism, but there's something about psychedelics in general. The second part that sort of supports this idea of the importance of the context is that if we gave MDMA to the animals, but we gave it in a social context, then we reopened the social critical period. But if we gave it in an isolation context, then we didn't. So what that is suggesting to us is that context is supervising which critical period is getting reopened. And so if we want to open a social critical period, we give a social context. But for example, we're thinking now that if we're trying to reopen a motor critical period, then maybe social context doesn't matter, and we need to switch to a motor context to get a motor critical period open. So those are ideas that we are testing right now.
Could you tell us why you got interested in psychedelics as a person?
Yeah, so when I was an undergrad, I was interested in psychedelics because I had seen this picture of the serotonin molecule sitting right next to the LSD molecule, and seeing the similarities in the molecular structure between these two compounds made me think that this is going to be how we were going to understand the brain, like sort of the big, important, interesting questions about consciousness and, you know, understanding how the brain programs itself to enable consciousness. And I thought this molecular similarity would give us the the leverage we needed to answer those big questions that seemed more in the purview of philosophy, for example.
How would you define what psychedelics are and what are the categories of psychedelics?
The definition of psychedelics that has been used for decades is that psychedelics are drugs that cause an altered state of consciousness. And so this large category includes drugs like LSD and magic mushrooms and DMT and ayahuasca and mescaline. Those are typically called the serotonin psychedelics, but it also includes in that category drugs like ibogaine, MDMA, and ketamine. And so the terminology that I favor is that while all of them are psychedelics, they can be subdivided based on similarities in the acute subjective effects. So for example, MDMA is different from other psychedelics in that it induces this empathogenic quality or this pro-social quality. Ibogaine is thought of as a onirogenic psychedelic because it induces a sort of dreamlike state. Ketamine and PCP, these are psychedelics that induce a dissociative state. And then LSD, psilocybin, mescaline, these are hallucinogenic psychedelics. So subcategories of psychedelics.
So if psychedelics open what we call the critical period, is it possible that one day people will use psychedelics for learning?
Right. So in principle, this idea of psychedelics being this master key for reopening critical periods would mean that we could return to this open state and essentially enable us to learn languages as an adult, or maybe become musicians as an adult if we missed our opportunity to learn those things as a child. But the danger is also that critical periods close for a reason. And the reason that most neuroscientists think they close is because it's energetically very costly to be in that open state all the time. I mean, if you've ever watched children, trying to go through a park, right? They're noticing everything. It takes them forever to do something that we as adults are able to do very quickly because we're relying on habits and learned efficiencies that those children who are trying to learn their environment are not capable of doing. So it's not only a matter of efficiency, it's also that, when you're in that vulnerable open state, you are much more suggestible, right? So the main historical example of a potential danger is the story of Charles Manson, right? So this is somebody who used psychedelics and gave psychedelics to people that he was trying to indoctrinate into his cult. And so he gave it to them several days in a row while indoctrinating them into this sort of social group that believed that he was, the Messiah, and turned a bunch of hippies into people who would, murder celebrities in order to achieve the helter-skelter mission of Charles Manson, right? So being in this sort of vulnerable state where you're suggestible, it's potentially very destabilizing and very dangerous. And so I think that's the thing that we always have to keep in mind, that these are very powerful medicines, and that what we're doing to people is restoring them to that state of vulnerability and suggestibility that we try and protect children when they're in that state.